Hello students, in Biozoology, fourth lesson, Principles of Inheritance and Variations. In this sex-linked inheritance, the inheritance of a trait that is determined by a gene located on one of the sex chromosomes is called sex-linked inheritance. Genes present on the differential region of X or Y chromosomes are called sex-linked gene. The genes present in the differential region of X chromosome are called X-linked genes. The X-linked genes have no correspondent allele in the Y chromosome. The gene present in the differential region of Y chromosome are called Y-linked or holandric genes. The Y-linked genes have no corresponding allele in X chromosome. The X-linked gene inherit along with Y chromosome and they phenotypically expressed only in the male sex because the males are hemizygous and therefore express the trait when they inherit one mutant allele. Hemizygous means it has X and Y. Next, inheritance of sex-linked genes. That a few characters are only occur in the X-linked genes. That red-green color blind or daltonism, hemophilia, and Duchenne muscular dystrophy are example of X-linked gene inheritance in humans. Hemophilia is commonly known as bleeder's disease. Hemophilia is caused by a recessive X-linked gene. A recessive gene for hemophilia lacks a normal clotting substance thromboblastin in blood. Hence, minor injuries causes continuous bleeding leading to death. The females are carrier of the disease and would transmit the disease to 50% of their sons even if the male parent is normal. Hemophilia follow the characteristic crisscross pattern of inheritance. That means that this gene is carried only in the X chromosome, that the females are only carrier of the gene that affects the male. Next one is color blindness. In human beings, dominant X-linked gene is necessary for the formation of color-sensitive cells in the cones. The recessive form of this gene is incapable of producing color-sensitive cone cells. That means dominant gene only responsible for the color, and the recessive is incapable of producing color sensitive cone cells. Homozygous recessive females and hemizygous recessive male are unable to distinguish red and green color. The inheritance of color blindness can be situated in the two types of marriages. It's an example: marriage between color blind man and normal visioned woman. A marriage between a color-blind man and a normal-visioned woman will produce normal-visioned male and a female individual in F1 generation. But the females are carriers. The marriage between a F1 normal-vision carrier woman means that woman has a color blindness in the cell, but she is a carrier, but she is not affected by that blindness. And a normal visioned male will produce one normal visioned female, one carrier female, one normal visioned male, and one color blind male. Color blind trait is inherited from the male parent to his grandson through carrier daughter, which is an example of crisscross pattern of inheritance. That diagram shows that type of Inheritance. Another example, marriage between normal visioned man and color-blind woman. If a color-blind woman marries a normal vision male, all F1 son will be color-blind and daughters will be normal visioned but are carriers. A color-blind male will produce normal vision carrier daughter, color-blind daughter, normal visioned son and a color-blind son in the F1 generation. This diagram also showing that color blindness, the marriage between normal vision man and color-blind woman. Inheritance of Y-linked genes. Few characters are only present in the Y chromosome. The gene in the non-homologous region of Y chromosome are inherited direct from male to male. In human, the Y-linked Hollandric genes for hypertrichosis, expressive development of hairs on pinna of the ear, are transmitted directly from father to son because male inherit the Y chromosome from the father. Female inherit only X chromosome from the father and are not affected. So this character is only transformed from Y chromosome, only in Y chromosome. Next one, karyotyping. Karyotyping is a technique through which a complete set of chromosomes is separated from a cell and the chromosomes are arranged in pairs. An ideogram refers to a diagrammatic representation of chromosomes. Preparation of karyotype. Preparation of karyotype. Tijo and Levin describe a simple method of culturing lymphocyte from the human blood. Mitosis is induced followed by the addition of colchicine to arrest cell division at the metaphase stage, and the suitable spread of metaphase chromosome is photographed. That means that during cell division in mitosis stage, colchicine is added to the cell, so the cell division stopped at metaphase stage, and after that, during metaphase stage the chromosome is photographed. The individual chromosomes are cut from the photograph and are arranged in the orderly fashion in homologous pairs. This arrangement is called a karyotype. The chromosome banding permits structural definitions and differentiation of chromosomes. The picture is showing human karyotype, that all the chromosomes are separated and photographed. Application of karyotyping. It helps in gender identification. It is used to detect the chromosomal aberration like deletion, duplication, translocation, non-disjunction of chromosomes. It helps to identify the abnormality of chromosomes like aneuploidy. It is also used in predicting the evolutionary relationship between species. Genetic disease in human being can be detected by this technique. Next is a human karyotype. Depending upon the position of centromere, chromosome consists centromere, that position of centromere and a relative length of two arms. Human chromosomes are of three types, that is metacentric, submetacentric and acrocentric. Pedigree analysis. Pedigree analysis is a family tree drawn with standard genetic symbol showing the inheritance pathway for specific phenotypic characters. Pedigree analysis is the study of triads as they have appeared in a given family for several past generations. There is a chart which is showing that the symbols used in pedigree chart, and this pedigree analysis, it has a genetic symbol which shows the inheritance from the generation to generation. The character is transformed. Next is a genetic disorder. Genetic disorder. A genetic disorder is a disease or syndrome that is caused by an abnormality in an individual DNA. Abnormality can range from a small mutation in a single gene to the addition or subtraction of an entire chromosome or even a set of chromosomes. There are two types of disorders, Mendelian disorder and chromosomal disorder. Mendelian disorder. Alternation or mutation in a single gene cause Mendelian disorder. If the disorder takes place only in single gene, that is called as Mendelian disorder. These disorders are transmitted to the offspring on the same line as the Mendelian pattern of inheritance. Example for Mendelian disorders are Thalassemia, Albinism, Phenylphotonuria, sickle cell anemia, Huntington chorea, etc. These are the disorders. It may be takes place in dominant or recessive and autosomal or sex-linked inheritance. That is sex-linked genes. Thalassemia is an autosomal recessive disorder. Autosomal means it takes place in body cell, and the recessive gene only disorder takes place. It is caused by gene mutation resulting in excessive destruction of RBC due to formation of abnormal hemoglobin molecules. Normally, hemoglobin is composed of 4 polypeptide chains, 2 alpha and 2 beta globin chains. Thalassemia patients have defect in either the alpha or beta globin chain, causing the production of abnormal hemoglobin molecule resulting in anemia. That if it is abnormality takes place, anemia is a result of that disease. In thalassemia, thalassemia is classified into alpha and beta based on which chain of hemoglobin molecule is affected. It is controlled by two closely linked genes, HBA1 and HBA2 on chromosome 16. Mutation or deletion of one or more of the 4 alpha gene allele causes alpha thalassemia. In beta thalassemia, production of beta globin chain is affected. It is controlled by a single gene, HBB on chromosome 11. So alpha is present in chromosome 16 and beta is present in chromosome 11. That gene. It is the most common type of thalassemia and is also known as Cooley's anemia. In this disorder, the alpha chain production is increased and damage the membrane of RBC. This is the disease. And the next one is phenylphotonuria. It is an inborn error of phenylalanine metabolism. That is, phenylalanine production is caused due to the pair of autosomal recessive gene. It is caused due to mutation in the gene PAH, that is phenylalanine hydrolase gene, located on chromosome 12 for the hepatic enzyme phenylalanine hydrolase. This enzyme is essential for the conversion of phenylalanine to tyrosine. This enzyme is essential for conversion of phenylalanine to tyrosine. Affected individual lack this enzyme, so phenylalanine accumulate and get converted to phenylpyruvic acid and other derivatives. It is characterized by several mental retardation, light pigmentation of skin and hair. Phenylpyruvic acid is extracted in the urine. In this disease, that phenylalanine metabolism is caused by autosomal recessive gene because this enzyme, the enzyme phenylalanine hydrolase is essential for conversion of phenylalanine to tyrosine, that is affected. So it is a phenylalanine is accumulated and converted to phenylpyruvic acid that cause the This is albinism. Albinism is an inborn error of metabolism caused due to an autosomal recessive gene. Melanin pigment is responsible for skin color. Absence of melanin result in a condition called albinism. Absence of melanin is called albinism. A person with the recessive allele lacks the triosinase enzyme system. So, if the person doesn't have triosinase, lack triosinase, which is required for the conversion of dihydroxyphenylalanine into melanin pigment inside the melanocyte. In an albino, melanocytes are present in normal number in the skin, hair, iris, etc., but lack melanin pigment. This is about albinism, the lack of melanin, which is caused albinism. Next one is Huntington chorea. It is inherited as an autosomal dominant lethal gene in man. It is characterized by involuntary jerking of the body and progressive degeneration of nervous system, accompanied by gradual mental and physical deterioration. The patients with this disease usually die between the age of 35 and 40. Chromosomal abnormalities. Each human diploid 2 nucleus body cell has 46 chromosomes, that is 23 pairs. Chromosomal disorders are caused by error in the number or structure of chromosomes. Chromosomal anomalies usually occur when they are in error in cell division. Failure of chromatid to segregation during cell division, resulting in the gain or loss of one or more chromosome, is called aneuploidy. It is caused by non-disjunction of chromosome. During division, there is a segregation, failure of segregation of that chromosome, maybe gain or loss of the chromosome, which is called aneuploidy, and it is caused by non-disjunction of chromosomes. Group of sin and symptoms that occur together that characterize a particular abnormality is called a syndrome. In human, Down syndrome, Turner syndrome, Klumpfer syndrome, Pater syndrome are some of the examples of chromosomal disorders. It is two types: autosomal aneuploidy in human beings, and another one is allosomal abnormalities in human beings. Autosomal aneuploidy in human beings. Several autosomal aneuploids have been reported in human beings. Example for this example is Down syndrome, that is 21 trisomy, and Pater syndrome 13 trisomy. It is takes place only in the body cell, so it is called as autosomal aneuploidy. Down syndrome trisomy 21, trisomy condition of chromosome 21 result in Down syndrome. So chromosome 21 is there, trisomy means it forms the Down syndrome. It is characterized by severe, characteristic of that disease is severe mental retardation, defective development of the central nervous system, increased separation between the eyes, flattened nose, ears are malformed, mouth is constantly open and the tongue protrudes. These are the characteristics of Down syndrome disease. Another one is Pater syndrome. It's a trisomy 13, trisomy condition of chromosome 13. In chromosome 13, trisomy condition is occur. This is called the Pater syndrome. Meiotic non-disjunction is thought to be the cause for this chromosomal abnormality. It is characterized by multiple and severe body malformation as well as profound mental deficiency, small head with small eyes, cleft palate, malformation of the brain, and internal organs are some of the symptoms of this syndrome. Next one, allosomal abnormalities in human beings. Mitotic or meiotic non-disjunction of sex chromosomes causes allosomal abnormalities. It maybe takes place in mitotic or meiotic non-disjunction of sex chromosome. So this disease takes place in sex chromosome, that is called allosomal abnormalities. And Klumpfer syndrome and Turner syndromes are the example for this abnormality. Klumpfer syndrome, this genetic disorder is due to the presence of an additional copy of X chromosome resulting in a karyotype of 47 XXY. Person with this syndrome have 47 chromosomes. They are usually sterile males, tall, obese, with long limbs, a high-pitched voice, underdeveloped genitalia, and have feeble breast development. Turner syndrome, it takes place in females. The genetic disorder is due to the loss of a X chromosome resulting in a karyotype of 45X chromosome. Person with this syndrome have 45 chromosomes and are sterile females. The character of this disease is low stature, webbed neck, underdeveloped breast, rudimentary gonads, lack of menstrual cycle during puberty are the main symptom of this syndrome. This is about this lesson. So today we have seen that sex-linked inheritance, that few characters are present only in the X chromosome and few characters present only in the Y chromosome. And inheritance of X-linked genes example is Hemophilia, color blindness, and next one is the inheritance of Y-linked gene is phalandric gene. Karyotyping is a technique through which the complete set of chromosomes is separated from a cell and the chromosomes are arranged in pairs. And ideogram refers to the diagrammatic representation of the chromosome. Preparation of karyotyping by means of during mitotic division, adding that Golgi sign which is stop the division, and during metaphase stage the chromosomes are photographed. And application of karyotype is gender identification, detect the chromosomal abnormalities, identify the abnormalities of chromosomes, and predict the evolutionary relationship between species is also used through karyotyping. And genetic disease in human being can be detected. And pedigree analysis means pedigree is a family tree drawn with a standard genetic symbol shown in the inheritance path for specific phenotypic characters. Genetic disorders, there are two types of that. Genetic disorder means a disease or syndrome that is caused by an abnormality in the individual DNA. It may be Mendelian disorder or chromosomal disorder. Mendelian disorder means it takes place, abnormality takes place in a single gene, and there are few diseases: Thalassemia, Phenylphotonuria, Albinism, Huntington chorea. Chromosomal abnormalities, that is disorders takes place in the chromosomes during the cell division, and during cell division that gain or loss of the chromosome which is caused that abnormality, and it may be autosomal aneuploid in human being or allosomal abnormalities in human being. In autosomal aneuploid in human being there are two diseases: Down syndrome and Pater syndrome. Allosomal abnormalities in human beings there are two diseases: Klumpfer syndrome and Turner syndrome.